Hello, and thank you for taking the time to view this presentation on the microhabitat utilization by small fish communities in the Kabompo River. Brief introduction about myself. My name is Craig Rennie. I have completed my undergraduate and honours degree at Rhodes University, and I'm currently undertaking my master's degree at the same institution, looking at small fish communities in the Kabompo River. Below, I have listed some of my research interests, as well as platforms that you may use to contact me if you have any questions about today's presentation. Just a brief introduction to the Upper Zambezi. Well, it is located in northwestern Zambia and is the section of river from the source north of the town of Munilunga down to the Victoria Falls, which is not illustrated on this map. The Upper Zambezi has several large tributaries, some that arise from the Angolan highlands, and then the Kabompo River, today's focal area, with a catchment that is confined only to Zambia. To date, there are more than 120 fish species that are recorded to occur in the Upper Zambezi. Move into our focus area. The Kabompo catchment has an area of approximately 72,000 square kilometers, and the river runs for 1,000 river kilometers from its source on the watershed between the Democratic Republic of Congo and Zambia, all the way down to where it joins with the Upper Zambezi. There are several large tributaries in the Kabompo catchment, and over here we can see some examples of habitats and areas in the upper part of the catchment. However, in the middle reaches where this study took place, the Kabompo River is approximately 50 meters wide in the low water season. The middle and lower reaches, such as the area around Jivundu, are sandy bottom dominated, just like the main stem Zambezi. The Kabompo River has a relatively small fishery compared to other regions in Zambia, such as the Barotse floodplains or Kafue flats, with a large portion of catches being kept for personal consumption, especially the smaller fish species. There are several threats to biodiversity within the catchment. Some of these include large-scale mining. Some of the largest copper mines in Africa are located in the Kabompo catchment. There are also several small dams which have already been constructed within the catchment, and several others which are planned or currently under construction. Deforestation is also of concern, with areas being cleared for practices such as agriculture or the production of traditional charcoal, as well as for the logging of high-value timber species for international markets. There's also the threat of non-native species such as Nile tilapia, which seem to have established well in the system and have the ability to hybridize with some of the native species. Despite the threats I have just highlighted, conservation and management in the catchment is limited due to a lack of scientific data. The Zambezian Headwater ecoregion is classified as data deficient with a low quality of biological and taxonomic data due to a lack of published literature from the region. The Four Corners report provided good insight into fish species diversity in the Upper Zambezi, including the Kabompo. However, literature on the Kabompo River is largely confined to consultant reports relating to the damming and mining projects in the catchment, and therefore this data is inaccessible to the scientific community. Several ecological studies have been conducted in the Upper Zambezi, with a large focus on the main stem Zambezi River or in areas such as the Barotse floodplains, with the focus being largely on commercially important species such as the breams and serranochromids, as well as the large catfishes, and this has often left the smaller fish species, which are also important in the fishery, understudied. So therefore, the aim of the study was to record species presence and identify microhabitat preferences of small fish communities in the Kabompo River as indicators of ecological change. And the small fish communities were selected due to their importance in the fishery and general lack of knowledge about these species. The study took place around the area of Jivundu in the middle reaches of the Kabompo River. Sampling was conducted along a 25-kilometer stretch of river during the low water season from September to November in 2019. Now this section of river can be considered untouched as there is a large national park on the northern side of the river, as well as there are few large settlements, low population densities, and low fishing pressure in the area. The section of river around Jivundu hosts a variety of habitats, from rocky outcrops, sections of river with shallow sandy bottoms and dense marginal vegetation, as well as sections of river with a deep channel and also dense marginal vegetation. So these are the 4 habitat types we focused on for the assessments. In total, we sampled 123 sites across the 4 habitat types. And these 4 habitats were marginal Phragmites, a Vallisneria species, woody debris, as well as rocky reaches. And the N value under each habitat denotes the number of sites that were sampled for each habitat. Sampling sites were identified through reconnaissance trips up and down the river by boat. Sites were identified in reaches of the river that were shallow and contained numerous small sites. A large determinant of site suitability was safety, due to the abundant populations of Nile crocodile that live in the sample area, and therefore sites were largely sampled in sections of the river that were less than half a meter deep. Prior to sampling, sites were identified in each of the reaches to ensure that sites were at no less than 3 meters away from each other, and they were then sampled in an upstream order. Sites were sampled using backpack electrofishing, and the fish were regularly emptied into a 20-liter bucket of river water and allowed to recover. At each site, the sampling time was recorded, as well as several other characteristics, such as site depth, current velocity, and the substrate type. The fish species were identified to species level, and their total lengths recorded. For each of the 4 habitat types, diversity, evenness, and abundance were calculated. This was done using the Shannon-Wiener Diversity Index, Pilu's Evenness Index, and abundance was calculated as individuals per square meter. An ANOVA was used to test for significant differences within these values between the 4 habitat types, and if a significant difference was highlighted, a 2-key post-hoc test was performed. Habitat utilization was assessed using a conical correspondence analysis in the vegan package in R, based on presence-absence data. Following this, chi-squared tests for association were used to test for significant associations based on the CCA ordination. Well, onto the results. From the 123 sites that were sampled, at 4 sites, no fish species were recorded. We sampled a total of 45 fish species, which some of them are represented below, through 26 genera and 12 families. These include members of the family Mormyridae, Cyprinidae, Claridae, Cyclidae, as well as Masticemblidae. Now, the 45 species we caught represents approximately 40% of the known species diversity from the system. Cichlids were underrepresented in the sampling, and this is likely due to their ability to evade the electrofisher. If we look at the abundance, diversity, and evenness result, in the following graph, diversity and evenness are plotted along the axis on the left, while abundance is plotted against the axis on the right. Shannon Diversity Index was highest in woody habitats, followed by Phragmites, and both these habitats had significantly higher diversity indices than compared to Vallisneria. If we look at evenness, evenness was fairly similar across the 4 habitat types, with there being no significant difference between the 4 habitats. The same is seen in abundance. Abundance was highest in rocky habitats, and there was no significant difference in abundances between the 4 habitat types. Now, the differences in diversity between the 4 habitat types have previously been attributed to aspects such as habitat complexity. Although complexity was not measured in this study, habitats such as the woody habitats and the Phragmites appeared visually more complex than the other 2 habitat types, and may explain the differences in diversity between the habitats. However, this may also just be an aspect of sampling, where rocky habitats and Vallisneria were typically in areas with higher current velocities. For the purpose of this presentation, I've focused on 15 species for the conical correspondence analysis that had a frequency of occurrence of greater than 10%. Now, these are the species which are depicted in the current slide, and these species represent 6 families and 9 genera. If we look at the results of the conical correspondence analysis, the first 2 axes explained 81% of the data. We can see a large number of species grouped towards the woody habitats and Phragmites. We had Tilapia sparmani and Enteromius eutenia, grouped strongly with woody habitats. We had 3 species, Nanocarex multifasciatus, Interomius linear-maculatus, and Leucostracola johnstoni, grouped strongly with the Phragmites habitat. And then we had 3 species, Interomius radiatus, Interomius fasciatus, and Interomius kirsteni, grouping in between both of those. We had 2 species grouped strongly with the Vallisneria habitat, these being Nanocarex micropterus and Interomius neefi. We then had the Okavango suckermouth, Chiloglanus fasciatus, which grouped strongly with increased habitat current velocity. We have the cichlid Pharyngochromis acuticeps, which seem to show no significant association with any of the 4 main habitat types. And lastly, we can see Labiocylindricus grouped strongly with rocky habitats, as well as the Cynodontis macrostigma macrostoma complex and the Mormyrid Hippopotambris and Soji, and both the Cynodontis and the Mormyrid grouped more strongly with increased habitat depth. Visualize these results a little bit better and include the findings from the chi-squared analysis. We can see that the 2 species that grouped in the middle showed no significant association to any of the 4 habitats based on the chi-squared analysis. We get the following 3 species, the Cynodontis macrostigma macrostoma complex, Labiocylindricus, and the cichlid Pharyngochromis acuticeps, showing significant positive associations with rocky habitats. We then had the barb Interomius neefi, which showed a significant positive association with the Vallisneria habitat. Several Interomius species, Interomius kirsteni, Interomius linear-maculatus, Interomius fasciatus, showed strong, significant associations, positive associations with the marginal Phragmites. The Nanocarex multifasciatus showed a significant association with both Phragmites and woody habitats, and Interomius eutenia showed a significant association only with the woody habitat. Now, the species at the bottom showed no significant associations with any of the 4 habitat types. However, they did show strong associations with other habitat characteristics such as depth and current, with the 2 species, Nanocarex micropterus and Chiloglanus fasciatus, showing a strong association to habitats that had high current velocities. The Leucostracola johnstoni was on the opposite spectrum and preferred habitats that had low current velocities. The Mormyrid, Hippopotambris and Soji showed no significant habitat association, however was significantly associated with habitat depth. Therefore, from the study, it is clear that several small fish species showed significant positive associations not only with habitat, but with other aspects such as current velocity and habitat depth. Therefore, there is potential to use some of these species as indicators for change in the catchment. Species such as Interomius eutenia and Nanocarex multifasciatus that showed significant associations with woody habitat may be important in assessing the impact of deforestation and logging in the catchment. Other species such as Interomius kirsteni and Interomius fasciatus that showed significant associations with marginal Phragmites may be important in determining the impact of aspects of habitat loss through bush clearing or other changes that may occur in the marginal vegetation. Species that had strong associations with increased current velocity may be used to assess the impact of flow modification and damming in the catchment. Lastly, a species such as Labiocylindricus that had a strong association with rocky habitats may be important in assessing the impacts of aspects of sedimentation that may come about through flow modification or deforestation and bush clearing in the catchment. I would just like to thank the following institutions for funding, equipment, and logistical support, as well as several individuals for assistance in the field. If you have any questions, comments, or suggestions about the presentation or interested in the work we may be doing, please feel free to contact me on any of the platforms listed below. Thank you.